Thank you, Deborah, for the introduction. And thank you, Marianne, for the invitation, and to all the other organizers. So earlier this morning, our expert panel told us about how undergrads don't read anymore. I would say there's a pretty high probability that one of their loved ones read this children's book to them at some time. And if somebody has read Cat in the Hat, I'm sure many of you have already read this book. To me, this is my favorite children's book of all time, mostly because of Thing 1 and Thing 2. I mean, how can you not love Thing 1 and Thing 2? Now, why is Dr. Zeus relevant to our discussion today? Well, if you're sitting in this room, you're most likely working on what I would call a lead optimization problem. You're trying to convert a promising antibody hit into a commercial drug. And if you're not working on Thing 1, you're probably working on Thing 2, which is enabling greater molecular complexity in the life science industry. Now, all of us have seen some version of this graph before. So molecules are becoming increasingly complex. Health authorities are demanding more information and more rigor, and investors and bosses are demanding shorter timelines, and the budgets are becoming tighter. But fortunately, ATUM is here to rescue you. So ATUM is a US-based company. We're based in the Bay Area. We're completely employee-owned. The company is actually still run by 3 of the original 4 co-founders. In fact, 2 of the co-founders, Klaus and Sridhar, are at the conference here. We're 160 employees strong, and we have a rich history of scientific innovation and inventorship. Now, I think of this long and arduous road from DNA sequence to IND enabling activities as this progression of multiple steps. And in ATUM, we really specialize in everything immediately downstream of antibody discovery, right up to GMP manufacturing activities. And we've developed a lot of platforms over the years that enable the discovery, development, and manufacture of clinical molecules. And we've developed several tools along the way, but honestly, I think many of you don't care about our tools, so let me just skip the tools and just go straight to the results. And over the course of 23 years, we've designed and made a large number of genes that have resulted in the expression of over 51,000 unique proteins, anywhere from 5 mil to the 50-liter scale. And as of Friday, we've completed 456 cell line developments. And again, as of Friday, we've enabled 54 INDs. When I joined the company a month ago, that number was 52. And so there's a pretty high likelihood by the time I'm done with my talk, that might be up to 55. So to demonstrate our capabilities, instead of talking about the methods, I wanted to show you 5 specific case studies to demonstrate how and why we've been so successful. And my first case study falls in the Thing 1 category, which is a lead optimization example. And again, in terms of our roadmap, we're working immediately downstream of antibody discovery, but before cell line development. Now, we've all had experience that antibody discovery process yields hits and not commercial drugs, irrespective of the method you're using for discovery. And that's because we're all playing some version of this whack-a-mole game. And typically, we all know that if you increase the affinity of an antibody, you either break the antibody or you move the other desirable properties in the opposite direction of what you want. And so the way antibody discovery and lead optimization has typically worked over time, the traditional screen has been just trying to improve the affinity of the molecule and then hoping for the best. But unfortunately, hope is not a strategy. And the modern way to do lead optimization is essentially to do lab in the loop, where you're simultaneously optimizing all of the key parameters that you need for the biology and the developability and the manufacturability of your molecule. And to me, lab in the loop is really this perfect marriage of AI and machine learning and then wet lab activities to sort of confirm those models, which is again consistent with what our expert panel discussed earlier this morning. So you start with a lead antibody sequence, you make variants, you express them, you build them, you test them, you generate data, you put that back into your model, and then you go round and round until you're essentially ready to nominate your candidate selection molecule, and then proceed to cell line development. And one of the reasons we're able to do this is we have this Lightning HEC platform where we're able to go from DNA sequence information to purified protein in only 10 days. And then we're able to do high throughput developability screens with very small amounts of protein. And this is a really cool example from our partners at Lundbeck, showing you our lab in the loop capabilities. So our partners came to us with an antibody that had low affinity, low thermostability, low titer, high clearance, and high aggregation. So pretty much everything you don't want in an antibody. And let me show you how the lab in the loop worked. So this graph shows you the affinity in the top left panel. So the green X marks where we started, and the red box was the target. But there were also a number of other parameters, and I'm just showing you 3 of the parameters that we evaluated in the study. So polyreactivity, expression, and thermostability. And after the first round of optimization, you can see most of the antibody variants clustered around that starting point. In fact, many of the antibodies were actually had lower affinity than the starting molecule. But that information is very useful because when it gets put into the model and new variants are created, you can see that in the second round, the affinity started increasing. By the third round, there were already hits that met the affinity target. And by the fourth round, there were many more antibody sequences in the target window. But very importantly, if you look at polyreactivity, there are many molecules that again fell in that target window, expression increased, and thermostability increased. Now, the take-home message for me about this example, and we're very grateful for our Lundbeck partners for allowing us to present this information, is that, look, however you do this calculation, the amount of possible antibody sequences is something similar to the number of atoms in the observable universe. So there's obviously no way that you can ever sample that space. But by only making 365 molecular variants, so just 4 rounds and less than 100 variants per round, we were able to increase the affinity 3-fold and then increase all the other parameters the partner was looking to achieve. So a very, very cool example of the ability of doing lab in the loop. And our understanding is that this molecule is currently in clinical development. Okay, so moving on to thing 2, where we're going to talk about molecular complexity. We all know that molecules are becoming increasingly complex, and these are just some of the formats that many of us are working on at this point in time. In terms of our roadmap, we're moving a little bit more downstream, and the focus is really going to be here on our cell line development capabilities, where we utilize our leaping transposase and our proprietary CHO cell line, the MyCHO cell line. So let me just tell you a little bit about how we got here. So about 20 years ago, we had already established state-of-the-art codon optimization capabilities and gene synthesis abilities. And we also had the ability to do vector optimization to optimize for expression. And even 20 years ago, we had the ability to do protein engineering. And this allowed us to increase the expression of antibodies and other proteins of interest. 10 years ago, we developed our leaping transposase using the tools that I've described above. So we were able to engineer an enzyme that's able to insert copies of the gene of interest, typically an antibody heavy chain and light chain, into very transcriptionally active regions of the CHO genome, which allows for high expression and also the ability to modulate protein product quality attributes. And so today, putting all these tools together, we still do gene optimization, codon optimization, we still do vector optimization. We have now our proprietary CHO cell line. We make use very heavily of our leaping transposase. And so we're able to make very high quality pools expressing high titer and high product quality in a very short period of time. And so a lot of customers come to us because they want us to rescue their difficult to express antibodies. And I'm going to show you just a few examples of this. So here is one data set that's been previously presented before. So on the left-hand side is CHO pool titers of a competitor's CHO cell line development platform where different pools were made, but the maximum titer was only in the 2.5 to 3 gram per liter range. But using our platform, so our cell line, our transposase, our codon optimization, our vector optimization, here the partner was able to achieve titers 3-fold higher than the best pool using a competing technology. My next example is where a partner came to us after they had done a very expensive cell line development campaign somewhere else. The highest titer they could achieve in that cell line, which is a standard antibody, but a difficult to express antibody, was only in the 0.3 gram per liter range. And again, just making 5 pools, making them very quickly with our existing toolbox, we were able to generate pools with a titer 5-fold higher. And again, we were able to do this in a very short period of time using all of the tools in our toolbox. And another example I'm going to show you is a complex molecule. Again, a partner came to us. The highest titer that they could achieve after going somewhere else was in the 1.5 gram per liter range. And again, in this case, by making 6 pools where we played around with vector configuration and regulatory elements, we're again able to increase the titer 3-fold. So I do want to put a disclaimer up there because I've shown you a number of very low titers, and I don't want you to walk away thinking that we're not able to identify clones with high titers. So I just looked quite recently at a few recent cell line development campaigns, and across a number of different molecule formats, you can see that our unoptimized pools before any kind of process development and scale up are achieving titers in that 5 to 10 gram per liter range. And as you'd also expect to see, the clone titers are typically higher than the parental pool from which they were derived. So one thing we like to tell our partners is instead of going somewhere else and doing an expensive cell line development campaign and then not getting what you want and then coming back to us to try to rescue the expression, is we offer this rapid pool assessment where we can very quickly determine the manufacturing readiness of your molecule. We can provide gram amounts of protein in weeks. The product quality that you get from that pool is representative of the final clone. It's relatively inexpensive. There's no MTA involved. Sometimes it takes longer to actually negotiate the agreement than to actually do the work. And then once you have that pool, you can go straight into a cell line development campaign pretty quickly. So I've shown you an example of how we rescued an antibody that was not ready for clinical development. And I showed you a few case studies of how we rescued difficult to express molecules. But wouldn't it be great if we could solve the problem before it happened in the first place? And I'm sure many of you in this room have heard the upstream parable before. So allow me to give you a modified version of the upstream parable. So imagine we have 2 superheroes, upstream and downstream, who are just chillaxing by a river one day, noticing a number of baby antibodies drowning in the river. So what happens next? Well, downstream man jumps into the river to rescue the drowning baby antibodies. But no matter how quickly downstream man manages to rescue the baby antibodies, he quickly becomes overwhelmed with more baby antibodies that are drowning in the river. And so fortunately, our upstream hero, upstream woman, she runs upstream, identifies the problem, realizes there's some monster who's throwing premature babies into the river before they're ready to swim. So our version of Upstream Women, our superhero, is our new platform called Lock-in. And what is Lock-in? Well, I want to first preface this by saying that bispecific antibodies come in many different shapes and sizes, as shown in this very nice paper, this genetic paper from 2015. And bispecifics have many wonderful properties, but unfortunately, they come with some liabilities. One big one being the risk of near-epitope formation, but these molecules can also be prone to misassembly, aggregation, instability, short half-lives, and they can be difficult to express. And so we've developed a proprietary knob and hole IgG1-like bispecific platform, which is a 4-chain molecule which allows us to essentially bind to 2 targets at the same time. And we've engineered these Fab regions, which I'm showing here in pink and blue, that allow for unique light chain mispairing. And we're actually able to produce molecules in 2 different flavors. So you can see in this diagram, I'm showing you the Fab pairings on the pink Fab pairings on the knob side, but we can also put the blue Fab pairings on the knob side. And to show you how this would work for a hypothetical program, so imagine you come to us with 8 CD3 antibodies, and you come to us with 8 unique CD20 antibodies, we can mix and match these molecules very quickly and very easily for you. So what this would look like if you have, 8 antigen A binders, 8 antigen B binders, you can make a matrix of 64 unique molecules. But if you flip the orientation around, you can imagine we can make 128 unique versions of bispecific antibody pairs. Now I'm going to show you some actually data where we took 8 commercial antibodies and then we paired those 8 commercial antibodies essentially with themselves. And so what this looks like in terms of the matrix, you can see in how we paired adalimumab with itself, atezo with adalimumab, elo with atezolizumab, all the way to the end of the matrix of 64 possible combinations. But we were able to do this in both orientations. So again, with these starting 8 commercial antibodies, we could make 128 unique bispecific molecules. We could then use our Lightning HEC platform, where we're going from sequence information to purified protein in 10 days. And then we can use the purified protein from that Lightning HEC platform to perform the high throughput developability screen. And actually, quite amazingly, if you look at those 128 molecules, they all express in our Lightning HEC platform, so at least to a level that we were able to purify enough protein and characterize further. Looking at the amount of aggregation by SEC, you can see for the most part, most of these molecules show very low degrees of aggregation. There are some pairings there on the left-hand side where you do see some degree of high molecular weight formation. And then again, another important product quality attribute is the correct pairing of the knob and hole heavy chains. So in this assay, which is a reverse phase LC-MS assay, you can see for the most part, these molecules are pairing correctly. The next assay was to determine the correct pairing by mass spec. And so the first way we do that is to look at intact mass spec. And this is just obviously one of the 128 examples. You can see most of the molecule is has the correct mass of the heterodimer. There's a little bit of homodimer formation there on the right-hand side. But then we also followed that up by doing a Fab digestion to release the 2 Fabs and then ensured correct light chain pairing by making sure that the 2 released Fabs correspond to the correct size. Now, to me, the most amazing thing is across this panel of 128 unique pairings, there was no detectable light chain mispairing involved. I've previously worked on bispecific cell line development at Eli Lilly and then at Genentech Roche, and I've never seen a case before where there was no light chain mispairing. So pretty remarkable that there's no light chain mispairing in this platform. We also wanted to make sure that the Fab pairings don't influence the binding. So again, looking at those 8 commercial molecules binding to their respective antigens, as you'd hope to see and expect to see, that the Fab pairings don't influence the binding. So the blue Fab pairing, the pink Fab pairing yields the same affinity as the wild type Fab pairing. And then one of the last characterization assays we did was to look at cell killing in a cell-based assay. So on the left-hand side, we have a lock-in version of CD3, CD20. And on the right-hand side, we have a genetic molecule as a control, which is mosin. And you can just see visually that in the cell-based killing assay that you see very similar profiles for the 2 molecules. What I really like about the cell-based killing assay example is that the target cells used for this assay is a leukemia cell line, the NELM6 cell line. It doesn't express a lot of CD20 on the surface. And so we actually used our leap-in tools to A, overexpress CD20 on the surface, and B, to overexpress the luciferase reporter gene. So again, showing you how all these different tools we have in our toolbox we're utilizing for the drug discovery and development process. And then lastly, we were able to make stable CHO pools using our leap-in platform and our MyCHO cell line. And in this case here, just 4 quick pools with a few different regulatory element combinations. You can see titers as high as 5 gram per liter in unoptimized pools. So again, also showing the molecule expresses well, and again, we can move very quickly from discovery all the way through to cell line development. So I hope to have convinced you today that our lock-in platform is a unique example of where we are working on thing 1 and thing 2 at the same time. We have a booth. It's actually a pretty cool corner booth, so it's easy to find. It's also double the size of what it was last year. We have many members of our commercial team here. They're all engineers and scientists. They all love to talk about science. So please feel free to stop by and chat with us. And thank you very much for your time and attention today.
Thank you, Gavin. Questions for Gavin?
What makes pink pink and blue blue?
It is essentially unique pairing sequences, right? So one of the sequences is CH3 and kappa constant domain, and then on the other side is a very similar type of structure, and we can talk about it after the fact. Yeah.
Okay.
Hi, Gavin. Great talk. Back here. Great talk. So 5 to 10 grams per liter is pretty impressive. Do you have a goal that we want to hit this number even far beyond 10?
I will answer that question by saying my vice president at Roche used to talk about the titer Olympics and how everybody was trying to outdo one another in terms of titer. But really the most impactful thing that we can do in drug discovery and development is essentially get into the clinic as quickly as possible. And I think it made a lot of sense to increase antibody titers in the early days of antibody manufacturing when titers were less than a gram per liter and you would need these like 6-pack farms of 20,000 liter reactors. But for the most part, when you are, I think 2 things are happening. Once you get titers in this 5-ish gram per liter range, that's typically more than enough that you need. certainly for clinical development and even for manufacturing. And then a lot of new drugs have really very small patient populations, or the molecules are becoming very potent. So you don't need a lot of kilograms anymore. So, I mean, yes, we're also trying to increase the titer, but we have much greater emphasis on speed. How can we enable the whole drug discovery and development process to go faster for our partners?
Other questions? I have one. If you can speak more to the different pools. So I know for the examples you had 5 pools. What controls that? And can you give any more color into how many a client could expect?
Yes. Well, the short version is it depends how much the client's willing to pay for, right? But I'm being somewhat facetious there. We have published on this before, so this is not proprietary information. So we have a library of different promoters. We have libraries of different, like 5' and 3' UTR sequences. We have different orientations that you can put the genes in. When we make 4-chain bispecific molecules, we typically have the genes split on 2 different plasmids, and you can play around with the plasmid ratio. So I think for the most part, with about 4, 5, or 6 combinations and pools, you can really get what you want. But we can obviously do more if that doesn't work, or if clients or partners want to do more along the way.
Okay, thank you. Any other questions? If not, we have a break right now from 10:35 to 11:15. So please go have coffee and view the posters, and then we'll see you back here at 11:15. And can we thank Gavin one more time, please?